Good morning, everyone. This is Nicole Ali. There is another article from The Atlantic. It was published a little while ago. I just hadn't gotten to it. If you can see in the title, this is a Sciencetalk article. I know I've put out a lot over the past week or so, or past couple of weeks. I had to come here because there are some things I'm studying concerning transposon, CRISPR-Cas9 technologies, and similar phenomenon occurring within the human genome. Some of them include RNA binding proteins as well as microRNAs. So this article is entitled, "The Mysterious Jumping Gene that Appears 500,000 Times in Human DNA." The image you see is a cell that's dividing in two with its DNA stained in blue. So this is a process called mitosis as well as meiosis that occurs later, where you see the centrioles that are stained in a yellow color in the middle here, and you have the blue DNA within the nucleus, and the rest including the cytoplasm in red and orangey red color. This was published June 21st, 2018. Its segments make up 17% of our genome. So the segments of the jumping genes, the transposons, make up 17% of our genome, which is quite a lot. But scientists are only just starting to understand what these jumping genes do. This is a pretty long article. I'm just going to describe the important parts. They start off saying here, for years, Miguel Ramalho Santos tried to convince researchers in his lab to study a segment of DNA he personally thought was quite extraordinary. He called it LINE1. It's repeated 500,000 times in the human genome, making up nearly a fifth of the DNA in every cell. But nobody in his lab wanted to study it. He says it was a sort of running joke in the lab. He's a developmental biologist at the University of California in San Francisco. It might have had something to do with LINE's reputation. People have called it junk DNA. So junk DNA was originally called introns. So the introns are pieces of DNA between open reading frames or exons. And for a while, people thought they had no function. People have called it genomic parasites. LINE1, like other transposable elements or jumping genes, has the unusual ability to copy and insert itself in random places in the genome. And that's why they considered it a parasite, because they didn't know its function at that time. Geneticists tend to pay attention when LINE1 inserts itself in a bad place, causing cancer or genetic disorders like hemophilia. As you know, those of us of the melanated community do not normally deal with hemophilia. This is a disorder that affects more of the high IQ individuals, those of the Caucasoid persuasion. But Ramalho Santos suspected there was more to LINE1. If LINE1 were at best harmless and at worst harmful, why would it persist and in such abundance in the human genome? And that's an interesting question to ask, because if it is, if it is harmful, the body or the genome would get rid of it, because in nature, there's a high conservation of energy. Number one law of nature is self-preservation. And in nature, the second law, right under self-preservation, is energy conservation. So if this is a gene that's in such abundance, it must have some function. And that's, I think, what he wanted to study. And here he had a hunch. He and others have found that LINE1 is especially active inside developing embryos, which suggests that the segment actually plays a key role in coordinating the development of cells in an embryo. He began doing experiments himself here and there, occasionally bugging a researcher in his lab for help with the analysis. They all thought it was a bit cuckoo, says Ramalho Santos. It wasn't until Michel Perchard joined the lab as a postdoc, and he finally found someone to take on the LINE1 project in earnest. She says, I think I got strange looks from the rest of the lab, and she recalled to the interviewer with a laugh. But the experiment started to pay off. Crucially, the two researchers figured out how to turn LINE1 off in mouse embryos. So they probably used CRISPR. They have a link here, probably for their study. They probably just knocked it out. So DNA stores the code of life, but to read it, cells have to first transcribe the DNA sequence into RNA. When the team neutralized LINE1 RNA, the embryos got stuck in the 2-cell stage right after a fertilized egg has first split. Without LINE1, embryos essentially stop developing. They stop dividing. Ramalho Santos thinks that LINE1 RNA particles act as a molecular glue, bringing together a suite of molecules that switch off the 2-cell stage and kick it into the next stage of development. In particular, it turns off a gene called DUX, which is active in the 2-cell stage. Over the past few years, other research groups have stumbled upon hints of LINE1's functions. They've uncovered possible roles in early mouse embryos and in brain cells, but they've never quite established a solid case for LINE1's functions. Until really recently, this search has been elusive. I think this is something we've all been waiting for, says Wenfeng An, a LINE1 researcher at South Dakota State University. It's a groundbreaking study. So they're finding that when they block LINE1 from inserting itself into the genome, the embryonic stem cells looked fine. So they had the normal phenotype, and it was also this gene was also preventing harmful jumping around in the genome. Then they looked at LINE1 in human cells, where it makes up 17% of the genome, and they found there were differences between mice and humans, which is expected. But LINE1 is abundant in the genome of almost all mammals. Other transposons, excuse me for the background noise, there's someone who has a crazy thing on their car. All right, let me just let them do what they have to do. Okay. Other transposons, also once considered junk DNA, as I spoke of earlier, have turned out to have critical roles in development in human cells too. Another person, Joanna Y. Saka, says there's a lot of treasure in the junk. She's also studied LINE1 at Stanford. If nothing else, Ramalho Santos may now have an easier time of convincing students to study his favorite, underappreciated DNA segment. They're trying to make this a joke, like, oh my gosh, he found this gene, and now he gets to study this gene. Oh, I'm so glad that he's able to do this. The more important thing to note is that they're going to start perhaps doing analysis with various ethnic groups as samples to compare the expression of LINE1 between Caucasoids and melanoid people. And when that comes, you can begin to determine the base function of the LINE1 gene. You can begin knocking it out, you can begin overexpressing it, and doing a lot of other manipulation genetically. I don't know what this means for the future, but if it is a harmful gene that is overexpressed in cancer or disease states, it may be something that they can use during stem cell research or regenerative medicine. I have to keep an eye on this and other genes like it to see what is to come of it. I hope you enjoyed this video. It is a science talk, and it may lead you to other discoveries or information that may be critical to you. Anyway, I will leave a link to this article in the description box, and I'll talk to you soon. Have a great day.